Okay, good morning. So this is the start of our last week of material for this semester. So today, Wednesday, Friday are the last three lectures. And then next week, of course, we have our third exam, and then that wraps up the course. So this week in recitation, you have a homework assignment due, and you will also have a quiz. You will have one more homework assignment, right, which will be assigned at the end of this week that will cover the material that we are talking about this week in lecture. And so that one will be due at the end of next week after you've taken the third exam. Right? You know how I always feel about the homework is, it gives you an opportunity to really think about these concepts, work with them, and importantly, get you more comfortable recognizing what the important points are that I expect you to be able to demonstrate at exam time, right? So in situations like this where you have the homework due after the exam, of course, I encourage you to try to do the homework or at least take a good look at the homework before the exam date so that you can benefit, right, from being from having the homework assignment, and so you know what to study. But then, of course, you don't have to actually finish doing the homework until you turn it in at the end of next week. All right. Okay. So let's talk about the cytoskeletal systems inside of cells. When we think about a cell, as we've been learning all semester long about the different types of components that make up the structures of the cells, we focused a lot on the actual macromolecules, things like lipids or proteins that make up different types of membranes. And we've already studied examples, very important examples of different types of membrane structures inside of the cell. Everything from the endoplasmic reticulum and the Golgi to different things like the membranes around mitochondria, right, that perform important functions like allowing for electron transport chains to set up proton gradients that power things like the ATP synthase proteins that are embedded in those membranes. I think one thing that we haven't really appreciated, though, is that all of these complex structures, organelles inside of the cell, it's easy to imagine them just kind of floating around, kind of bumping into each other, right? If we imagine the cell as this very fluid, basically sac of mainly water, proteins, there's carbohydrates, nucleic acids, bound up by lipid membranes, right? It's easy to imagine this as just like a sac of biological material, and all the structures inside are just kind of moving around. Like if you took a cell and shook it around, right? You can imagine everything inside just kind of like floating around. So that, of course, is not accurate, right? The different structures, the organelles inside of the cell, are not just floating around. They're actually attached to a very intricate three-dimensional network of different protein fibers that keep things sort of suspended and spaced apart the way they should be. In addition, these protein fibers also provide the means or the mechanism by which things can be moved around intentionally inside of the cell. Again, right, the life functions of the cell would not be very efficient if the cell, all it could do is sit around and wait for things to randomly bump into each other inside. I think you all know that by now, and it can appreciate that by now, right? In order for life functions to happen, in order for all of these wonderfully complicated biological molecules to interact at the right time and at the right place, things have to be moved around on purpose in order to encourage things to happen much faster than ordinarily they'd be allowed to if things were just encountering each other by chance. So let's look at this cartoon here on the left. Right, so this is how we've been sort of picturing a typical eukaryotic cell all semester long. We have these familiar membrane-bound structures, like, for example, in orange here, the endoplasmic reticulum. Here in pink is the Golgi, and in blue are the mitochondria, right? Again, these are not just floating around, crashing into each other, right? They are actually held in place by a three-dimensional network of fiber structures. So here, if we look at this microscope image, we can see many of those organelles in there. Right? Certainly, we can make out the appearance of the nucleus and all of these funny squiggling things. Maybe these are mitochondria, for example. Lots of vesicles, lots of membrane structures sort of cut in cross-section. Right? Let's zoom in. If we were to take a little area of this and really zoom in, we'd realize that this space is not just water with things floating in it, right? There is this complicated network of protein fibers. And so there are three main protein fiber types that make up the cytoskeleton of the cell. Here, very nicely, they've been stained in different colors so that you can kind of see that there are three different fiber types here. In pink, these thick ones are the microtubules. In blue, these sort of next thinner ones are these intermediate fibers. And then the thinnest ones, the ones that are sort of golden colored in this image here, are the actin microfilaments. Now, of course, in real life, they would not be these colors, right? In real life, pretty much none of the proteins or structures inside of the cell would have any color, because after all, they are just proteins and lipids, right? But when you're able to go ahead and use things like antibodies to label specific protein types, you can then have these sort of false color images produced. Right? Notice that there are these funny sort of gray little blobs that are attached to various fibers in here. What are these, right? Is that this junk? No, those are large protein complexes that are being used by the cell to carry out different types of biochemical life pathways. Notice how they're attached to these filaments. Right? In fact, I think none of the blue blob or gray blobs in this image are just floating around by themselves. So this really gives you a sense, right, that all these important components, even down to the level of big complex protein structures, are being moved around on this network of different fibers. So these fibers then, these three types of fibers, are responsible for things like structural support of the cell, so the shape of the cell. Right? Otherwise, if you think about this, right, if there's nothing really holding up the cell, and it's really just this lipid encased, blob of proteins and water and lipids, a cell wouldn't have much shape, right? It'd be no different than if you put a drop of oil on the counter of your kitchen, right? It would just kind of just spread out, and it would have no real structure, no real shape. So the shape of the cell is held together by the cytoskeleton, either keeping the shape of the cell intact if it's a particular cell that has to maintain a rigid structure or shape, right? So statically holding it that way, or certain types of cells, right, can change their shape as a function of them doing their normal cell functions, right? So the cytoskeleton can also be very dynamic and contribute to the intentional changing of the cell shape. Also, they are responsible for number 2, positioning of cellular components inside. That's what we just talked about. And number 3, also something that we just mentioned, is that these cytoskeletal fibers are very important for intracellular transport and trafficking. So how extensive is this cytoskeletal network? Right? I think oftentimes when you see illustrations of cells, like the cartoon on the previous slide, they don't bother indicating the cytoskeleton, because quite frankly, the cytoskeleton, if you drew all the fibers in there, it would just cover up everything in the cartoon, and you wouldn't be able to see the organelles that maybe a particular cartoon is meant to show you, right? But let's not forget that there is this extensive network. We can look at this and appreciate that by looking at these cancer cells. So these are HeLa cancer cells, viewed under typical light microscope. This is maybe what you would see under a microscope if you've stained a slide of cells with like H&E stain, for example. So just with these stains, you can see, for example, the boundaries of the nucleus or the nuclei, right? So these big sort of purple things are the nuclei. These are cancer cells, so they have really messed up genomes inside of them, right? And you can barely make out these sort of wispy sort of appearance of the cytoskeleton surrounding each of these cells. Now, each of these cells, of course, has one nucleus. So in this region in the middle here, maybe we have like 4 cells, 5 cells together, and you can just barely tell where one cell ends and the next begins. Again, because most of the cytoplasm is clear, right? So you cannot really see the boundaries of the cells. What if, instead of doing H&E staining like you would do for a light microscope like this, what if we were to go ahead and stain with, let's say, particular antibodies that bind to the cytoskeleton? What would it look like then? Well, these HeLa cells would look very different. They might appear like this. So in this case, we are fluorescently staining for the presence of microtubule filaments in green. The nuclei are still stained in blue, right? So you can see the big nuclei. But now you can really see the cytoskeleton of each one of these cells, and you get a better sense where one cell begins and the other ends, right? Where they're pressed up against each other. Notice also that they are staining in this image in red for the different proteins of the Golgi, right? That extensive membrane network that's used to package proteins that are newly made, right? And importantly, to begin trafficking or sending those proteins out to the different parts of the cells where those newly made proteins are meant to function. Notice that the microtubule network is extensively integrated with the Golgi. Right? So remember, I gave you that analogy before, that if the Golgi is like the shipping department of a factory where you're making stuff, making products in the front part of the factory, right? Like that might be the ER, and then things need to be packaged up in the Golgi before they are then loaded up on trucks, for example, and then shipped out to different destinations, right? Different maybe distribution centers. So it would make sense then that if things are going to get shipped along different sort of directions to go to different parts of the cell, right? Yes, of course. Right? These proteins, these products can't just be released and float around in the cell and you just hope that the proteins get to the right place in the cell. They have to be transported, trafficked along the cytoskeleton. And that's why you can already get this impression that you have extensive microtubule networks coming out from the Golgi on which different protein products are going to be moved. Okay. So we mentioned that there are 3 main protein fiber types that make up these cytoskeletal fibers. They are the microtubules, the microfilaments, and the intermediate filaments. Those are the 3 main cytoskeletal fibers. So we're going to spend most of today's lecture just looking at how these fibers differ from each other, right? Not only in construction, but also things like function. Okay? Not surprisingly, this is another example of how when life on Earth first evolved and simple cells first evolved, right, they needed to come up with these sort of fibers to be able to do things, right, like keep the cell the right shape, to be able to move things around inside of the cell. And so there were 3 main types of fibers that were sort of invented by Mother Nature. And it's not surprising then that some of the functions of these fibers overlap. Let's look at this here. So let's start off with microtubules on the left. Microtubules are essentially a hollow tube made up of a series of these protofilaments. Okay, so all 3 of these types of cytoskeletal fibers, right, are made up of protein units, right? But the way that these protein units, the identity of them and how they're arranged is what gives them their different structure. So you can see here in the microtubules, you have these alternating monomers, monomeric protein units of alpha and beta tubulin. So you end up making this hollow tube, which is typically 13 of these protofilaments sort of together to form a tube. And the diameter of that tube is about 25 nanometers, right? So it is the largest, right, or widest of the three cytoskeletal fibers. Okay, let's take a closer look at how these microtubules are made and why they are important. So if we start from the middle of this figure here, so we're looking at the microtubule or MT structure, a single unit might be thought of as a dimer, an alpha tubulin protein and a beta tubulin protein coming together as a dimer. So on the right are the ribbon diagram structures. You can see the alpha tubulin and the beta tubulin are highly similar in structure. So certain variations in amino acids make a slightly different structure, but very clearly both of these are tubulin molecules. When they bind GTP, or guanosine triphosphate, that changes their shape into the right shape, where two of these units can buckle together, an alpha and a beta. Okay. And then likewise, if the alpha appears bound to a GTP, then on top of this would be the next dimer of alpha and beta buckling on top of that. So if you continue that, you end up with one individual filament. Right? So one of these alternating red, yellow, red, yellow, red, yellow, right? And you'd have 13 of those in a ring to give you the full microtubule. Okay. Hollow tube inside. So if we go back to the cartoon on the left, you can appreciate the structure again, right? That there are 13 of these protofilaments sort of wrapped around to create a 25 nanometer diameter tube. Also important is this tube has a polarity, right? Because one end of it would always be where you end up with beta tubulins, sort of at the very end. The other end of it, you would end up always having alpha tubulins at the end. Okay. And so we call this the plus end, where you have the beta tubulin ending, and the minus end, where you have the alpha tubulin ending this tube. Why is that important? It's important because when tubules grow and shrink, this plus and minus end sort of dictates the direction in which the growing and shrinking takes place. You can see in this illustration, microtubules grow from the plus end. So in this top illustration here, you have a microtubule of some length already, right? So the plus end would be the end where the beta tubulins are sort of ending over here. This is where the next dimers of these alpha and beta tubulins get added on. So in this illustration, they're just making them blue colored so that you can keep track that these get added to the plus end. The other end is where the dimers are being taken off, right? So the minus end. So if the minus end was sort of stable and not being taken apart, and you just kept adding things onto the plus end, then you can imagine this microtubule growing or elongating. Okay. However, if the rate at which, let's say, dimers are being added onto the plus end is more or less equal to the rate at which the dimers are being taken off the minus end, then what would happen? Right? The microtubule would essentially be moving. Right? So you could appreciate that as these blue dimers get added, eventually, right, they would end up in the middle, and you can imagine them kind of moving to the left in this as this tubule is kind of being moving in one direction there. Right? And importantly, when it comes time for the tubule to shorten, right, the shortening is a function of the minus end being taken apart. So where do these microtubules come from inside of the cell? So they come from specialized protein structures that we refer to as microtubule organizing centers, or MTOCs. So a couple of examples of these MTOCs are things like the centrosome and also basal bodies. Right? So a centrosome, remember the centrosome is involved during mitosis, because the centrosome, when you have two centrosomes, they define the two sides of the dividing cell, right, to which the two sets of the genomes get pulled so that you can end up with two identical daughter cells. During interphase of the cell, remember interphase is basically all the other phases of the cell other than mitosis, Remember, the chromatin is pretty loosely organized, right? You would have a centrosome, and the centrosome, if you look at the structure up here, right, it's made up of, so the centrosome is this protein structure from which these microtubules are emanating from or growing from. So all of these microtubules that are growing out from the centrosome, the plus end, right, is the end that sticks out, because this is the end where additional tubulin dimers get added to. So if you look at this here, right, tubulin dimers are being added, so this fiber grows. The other end, the minus end, is held in place, anchored very firmly by other types of tubulin proteins, like, for example, this green gamma tubulin, right, and these other brown proteins that are sort of anchoring this tubule there. So once the cell enters mitosis, you have two sets of centrosomes now, and these centrosomes begin to migrate to either side of the cell. Importantly, what's going on inside the nucleus, now during prophase, you have the chromosomes beginning to really condense now, right, in preparation for being divided up, right, or allocated to either side. During metaphase, now the chromosomes are tightly condensed as they can be. This is when they are in that familiar sort of stubby X shape that I think most people, when they think of chromosomes, they think they're like this. Remember that for convenience sake, the textbook illustrations, and therefore our lecture illustrations, also to keep things simple, we pretend that the human genome, instead of being 23 pairs of chromosomes, right, we're just pretending like we just have 2 pairs of chromosomes, right? A big pair and a small pair, right? We said last time, let's just pretend it's like chromosome 1, because that's the largest one, and maybe chromosome like 17 or 18, one of the smaller ones, right? But recall, you still have one big chromosome from mom, one big chromosome from dad, right? And you still have one small chromosome from mom and one small chromosome from dad, right? During metaphase, right, the duplicated chromosomes, because each one of these duplicated during S phase, so when they condensed, you have the 2 sister chromatids that are still attached to each other. The microtubules from the centrosomes are going to grow outward, right, and attached to the kinetochore proteins on the center of the chromosomes. Right? And this is now creating what we call the spindle fibers. So you can imagine this dynamic process where the microtubules are growing, right, because you have more and more tubulin dimers being added to the plus end, and the plus ends eventually attach to the sister chromatids, right? And then when it's time for the spindle fibers to retract and pull the chromatids apart, how does that work? Remember, the microtubules, in order to get shorter, they get taken apart from the minus end. So essentially back at the centrosome, right, these dimers that are anchoring here are being taken off. So as these are being taken off, right away, the next set of dimers still remain attached. Right? So that's how the microtubules shrink away and physically pull the sister chromatids apart, allocating them to either side of the dividing cell. Okay. So microtubules then can have various functions. You can have cytosolic microtubules that dynamically grow and shrink. So these would be the ones like we just looked at during mitosis. They make up the spindle fiber. They organize and maintain cell shape, polarity. They're also responsible for the chromosomal movements during mitosis. And importantly, right, these microtubules can serve as sort of highways or different roads within the cell on which intracellular trafficking and transport can happen. In these wonderful, colorful illustrations here of different cell types where microtubules have been stained with different fluorescent markers, I think these are beautiful, right? Like the ones on the left, these green ones, you can really appreciate how these green microtubule fibers define the shape of the cell. Here you can see cells that are, maybe this is in the process of dividing. So now you have 2 daughter cells that have finished or finishing cytokinesis, right? So they're still stuck together by microtubules, keeping them together until they can fully split apart. Here's another beautiful image of microtubules in yellow here as being this extensive network, maybe to transport things inside of the cell. This one over here is nice because you can really see these little purple specks might be a particular set of protein complexes that are being transported around on these green stained microtubules. In addition, microtubules, because of their strength, you have certain microtubules that are stable. They don't grow and shrink. They're not very dynamic, right? So these are the ones that form different types of cell structures on the surface, things like cilia, right? So this might be an epithelial cell in your respiratory tract. And so the epithelium has these cilia. If you were to zoom in, you would see these are essentially, excuse me, bundles of microtubules together. If you were to take a cross section, you would see something like this, right? Where each one of these little round things is a microtubule. And you can see it's bunched together to give a very strong structure, right? So things like flagella, like the little bacteria that flap around the flagella like a little tail, right, to move them around. Those are examples of microtubules. Okay. Let's move to the next example, microfilaments. Okay. So microfilaments are the smallest diameter of these three fibers, typically 7 nanometers. They're also made of proteins, but in this case, they're made up of individual monomers of one type, known as G-actins. So you can see here, these little individual G-actin proteins, right? They're strung together. This kind of looks like a pearl necklace, right? Intertwined, 2 of these fibers. Right? So let's take a close look at these. So microfilaments are made of actin proteins. So an individual G-actin is a globular protein. So this blue structure on the left is the ribbon diagram of one individual monomeric unit. And you can see it has certain features that we'll discuss. One is it can bind ATP in the middle, right? So here's adenosine triphosphate in here. I also want you to notice that this is one individual protein. So they've labeled the N-terminal end of the protein here, right? Because if you were to unwind this would just be one long polypeptide chain of amino acid residues, right? From the amino terminal end, the very first amino acid, if you were to follow this around, sort of like a maze, like a game, right? If you follow this around, it would eventually end up winding around. And then here is the C-terminus of it, right? Another wonderful example of how the amino acid sequence of this will give it this particular shape when it folds up because of all the side chains of the different amino acids trying to get comfortable, right? The other thing I want to point out to you is that up here, they call this the pointed end of the molecule, and this part down here, the barbed end. It's kind of hard to tell, but you can kind of imagine that this part of the protein up here gives it kind of a point on the top. Right? It's the highest most point in this illustration here, whereas the bottom is much more spread out. Right? It's what we call the barbed end. If you look at this cartoon over here, you can see these are the individual G-actins. The little blue is the ATP that's bound in the molecule. When they bind ATP, right, the monomers can come together, and then when the ATP is converted to ADP, that changes the shape of the monomer a little bit so that it then holds on tightly to the next monomer next to it, and you form this chain. Okay. Here it is with the space filling model, a bit more realistic, and you can see how you can almost sense that it's twisting a little bit, right? The shape of these individual monomers are twisting around as these 2 fibers are intertwined. If you have a high resolution electron micrograph, you'll see here they are here. In this illustration, you can begin to see why there's a barbed end and a pointed end, right? Because you can tell there's almost this sharp kind of appearance here. I think of it as sort of like a molecular baggy tie. You are all familiar with these baggy ties, right? That you can use to tie up, for example, a bag of trash to take it out to the trash bin, right? Why are they barbed like this? Because the barbing allows, if things are being pulled against them, right? You can pull things one way, but it won't go the other way, right? In a baggy tie, that's how it stays together. Right? But these microfilaments are designed the same way. Because they are barbed, this gives them sort of a directionality, right? You can almost imagine that it can be sort of like scooted or shifted towards the pointy direction, but it would sort of catch and not be able to pull back. Likewise, other proteins that might be moving on these filaments can sort of hang on and catch on to the barbed nature of it and move things in one direction. So we can really appreciate that in this example here. So let's look at this particular cell. So this is a cell that's kind of spreading out, and maybe it's moving from the left side to the right side, right? And so you can actually see the cytoplasm of the cell spreading out. And you can almost sense that these sort of extensions of the cell membrane are grabbing onto the surface, right? And the rest of the cell is kind of going to get hauled along. So these microfilament, the barbed nature of it makes these filaments very useful for things moving against other things. That's why the filaments that make up our skeletal muscle cells, right, are these actin microfilaments, right? That's why when they catch on each other, you're able to actually contract your muscles, right? And then when you release them, you release them. You release your muscles. So they're important for cell locomotion and cytoplasmic streaming, meaning as the cell's moving forward in this image here, you can imagine the rest of the cytoplasm with all the proteins kind of making use of the barbed nature of these microfilaments to kind of move forward. So if we were to look inside of this cell, there would be these microfilaments all throughout, but the arrangement of these microfilaments might be different depending on what part of the cell we're looking at. For example, in this part on the left, the microfilaments may be arranged more in sort of this sort of roughly parallel orientation. But if you look down here, right, the actual direction or the barbed sort of pointiness might be just sort of back and forth. What would this do? This just creates, right, a good strong kind of support for the cell in that area. As you move closer to the region that's kind of reaching forward as the cell is moving, you might find, for example, as you get closer to the lamellipodium, right, you'd have them beginning to be more oriented in one direction. Right? Once you get out to the leading edge of one of these extensions of the cytoplasm, you might find that the microfilaments, right, are tightly parallel, and importantly, they're all arranged in one direction, right, so that the rest of the cell, as it flows over this, right, it can kind of hang on to the barbed nature of these filaments and move forward. Right, so you can see that. So let's zoom in, pretend like we're zooming in on these two little regions here, the lamellipodium and the leading edge, right? And this is what you would see. Right? In the lamellipodium region, you'd have these microfilaments sort of crisscrossing, and they're held together, sort of like buckled together or stapled together by other proteins, like these little filaments that are these little blue sort of like staples holding them together. In the extension of the leading edge, you can really sense that this is a tight sort of bundle of parallel microfilaments, right? They're tightly parallel, and they're held together again by things like fibrin, in this case, the little purple connecting protein. The third group of cytoskeletal fibers that we'll talk about in this class are the intermediate filaments. So you can sort of see a theme here. They're called intermediate because their size, their diameter is sort of in between the microfilaments and the microtubules, right? So these intermediate filaments, typically 8 to 12 nanometers in diameter. So they're also made of proteins, because all three of these are made up of proteins, but these are a little bit more variable. So you can see here, these are made of two different sort of individual lengths of protein that are sort of twisted together already, right, to form an individual dimer or an intermediate filament dimer. Right? The identity of the particular proteins that make up these little dimers is more variable. Right? So it's not like the microtubules that are only made of alpha and beta tubulins, or the microfilaments that are only made up of G-actin monomers forming an F-actin string of pearls. Right? In this case, there are 6 different classes of proteins that all can contribute to make structures like these individual components of the intermediate filaments. So intermediate filaments provide strength. That's their purpose. So unlike the microtubules and the microfilaments, that can be a little bit more dynamic, and they can be assembled and taken apart sort of dynamically because they're made up of individual little dimers of globular proteins or individual little monomer proteins. The intermediate filaments, these are highly insoluble, right? They don't dissolve easily. They're not taken apart very easily. So therefore, they function as the underlying shape and real structure and strength, rigidity of a cell. So I mentioned that several different types of protein units can be put together to make, twisted together to make these little intermediate filament units. So vimentins, keratin, lamin, these are just 3 examples. Keratin, right, we've mentioned a few times during class, keratin is that tough, insoluble protein that your skin cells make. Your keratinocytes make a lot of keratin, right? So that after the skin cells die, the keratin, because it's insoluble, right, doesn't dissolve easily, that's left over. And that's what gives you your tough, impenetrable outer layer of skin. The hair around your head is largely keratin as well, right? And you all know when you wash your hair, it's not like your hair dissolves, right? It's very insoluble. And so here, let's look at the structure. So here we have a dimer of 2 of these intermediate filament proteins. Maybe it's keratins, maybe it's vimentins, twisted together. Right? And then these individually can come together, you can see, to form a very strong fiber. All right, so from a dimer, you get a tetrameric protofilament, and then these tetrameric protofilaments come together to form a protofilament, and then these individual protofilaments are then bunched together to make an intermediate filament. Right? So one way of thinking about it is that, you know, if you're familiar with like loofah gourds, maybe some of you, use the dried loofah as like a, I don't know, exfoliant or something like that, right? I think we're all familiar with seeing those. Right? Now, of course, the structure of the loofah gourd is very rigidly defined by this tough, insoluble sort of skeleton inside of it, right? That gives it its rigidity, it gives it its shape. Now, in this case, of course, these filaments are not made up of these different types of intermediate filaments like we're talking about here. In this case, these filaments inside of a loofah gourd are made out of cellulose, right? So they're actually a carbohydrate, not a protein. But the reason I use this example, right, is I want you to think about how intuitively having a tough, insoluble structure like this, right, is very important for the rigidity of something like an individual cell. Right? So don't get confused. Don't say Dr. Shen taught us that the dried loofah thing is actually intermediate filament proteins. It's not, right? But I'm just using it as an example so that you can appreciate how important intermediate filament protein fibers are inside of the cell to give it rigidity. So here is a human vimentin assembled as protofilaments, right? So you can really see they're kind of twisted together, and this gives it a lot of strength when you have a number of these now packed up together. Okay, this is a really nice illustration, right? So we've been talking about 3 main types of cytoskeletal fibers, right? We have the intermediate filaments, in this case, maybe made of vimentin, being stained in green here. Remember, the intermediate filaments can be made up of one of 6 different families of proteins. Right? Vimentin is just one of them. Keratin was another one. Here are tubulins making up the microtubules. Right? So you have your alpha and beta tubulin dimers making these up, and you have your microfilaments made out of actins, right? Stained in blue here. So if we look at this image down on the left, right, this really nice false color electron micrograph, you can see how these 3 filaments work together. So we have the intermediate filaments in green, right? These are the strongest structural support ones. So you can see this sort of forms like this almost like anchor around which things like the microtubules in orange sort of follow or are used to sort of lay on top of the intermediate filament network, right? To actually provide things like now different pathways to transport things. In blue, or in this case, sort of grayish blue, all of these are various actin filaments over here. Okay, so these actins are over here. Right? And you can see how they're connected. So in this case, plectin is another example of a protein that can connect the various cytoskeletal components. Right? So this cartoon on the right is really meant to serve as a key for you to understand what you're seeing on the left. And you can really appreciate how these three main fiber types, the intermediate filaments, the microfilaments, and the microtubules, right, all work together, and they're carefully coordinated by being connected by other types of proteins in between them. All right, so that brings us now to a discussion about how things actually get transported on these types of fibers. Because I think so far in this lecture, it's pretty easy to appreciate, yeah, I get it. They provide structural support. They provide the ability to actually space things out and keep things sort of spread out, suspended inside of the cell, right? So things aren't all just bunched together in one place. But how about this whole idea of things being moved along and along these fibers? So here again, another beautiful example of staining to look for these fibers. Let's look on the right here, right? In this cross section, you can see things like mitochondria, endoplasmic reticulum, they're all suspended, right? And not just floating around freely. Even things like the ribosomes. So on the surface of the rough endoplasmic reticulum, you see lots of little purple ribosomes, right? But there are ribosomes in the cytoplasm as well, right? And this allows you to see that if you did not have any of these fibers visible here, it would look like all this stuff is just floating around, right? You also have the microtubules passing through here, where things are going to be transported. So the cytoplasm of the cell, or the cytosol, it is a very busy place because you are making tons and tons of proteins. And then as you're making proteins and other types of cellular products, right, they are not just going to be allowed to float around haphazardly and you just kind of hope that things come together, right? Because life wouldn't work like that. So in this factory that's making products, right, it's not like you make products and you just chuck them aside and they end up just bunching up somewhere. It's not just static and random. It is a highly organized and dynamic place where as products are being made by the cell, proteins are being put together into big complexes, or things like other molecules are carrying nutrients around, right? Things are dynamically being shifted around all over the place, and it's highly organized. How do things move like this? Right? So if you have those cardboard boxes on the conveyor belt, right, that's easy for you to relate to, right? Things are just kind of rolling along a conveyor belt, or the conveyor belt is kind of moving. But how do things move around inside of the cell? The way things get moved around or transported along the cytoskeletal network involves protein complexes that attach on one side to a particular cargo that they are carrying, right? And then on the other side, attach to the particular cytoskeletal element. And these protein complexes end up actively transporting things along the cytoskeletal filament, or the cytoskeletal fiber. An important family of proteins that do these are called the kinesins. So from the name, you know, kinesin, kinetic, right? Here is the structure of a kinesin. So it's kind of funny. It looks like sort of this bug-eyed fish, right? So it's two kinesin molecules that are intertwined with each other. So each one has a globular domain represented in blue that can bind to ATP. Then you have this sort of more chained linear region, and then on the other end you have this other sort of blobby type structure of the protein, right, which are the light chains. And so here you have it shown here in a much more accurate 3 dimensions, right? So the globular regions that bind ATP are these ends, right? And then these are twisted together, and on the other end you have this pretty intricate 3-dimensional sort of light chain region. Now, obviously, in real life, again, the kinesin protein is not going to be blue and green and purple and gray, right? But they've colored it this way for you to appreciate these different functional parts. Really think about it as just each one of these So this is 2 kinesin 1 molecules, right, that have, as a result of their amino acid sequence, globular domains on either end and a more linear region in the middle, and twisted together, you get this wonderful shape, right? So you might say, well, this thing looks really funny, right? So what's this? This looks like a pretty intricate sort of attachment clamp, right? And that's exactly what it is. It is a clamp that can hold on to something, the cargo that this thing is going to carry. Right? The other end, look, it looks like these 2 funny sort of cartoony feet, right? This is the end that binds to the microtubule. And so here you can see, right, the kinesin here, this dimer, right? This end is attached to a cargo. Maybe this is like a vesicle or something like that. Right? And then the other end, the little feet, walk along this microtubule, right? So when I said it looks like cartoon little feet, I wasn't joking. Right? So these bind to ATP. So when the forward foot binds to ATP, you can see here, ATP is coming and binding here, that changes the shape of the foot, right? Because the protein has to shift around a little bit, and there's a ripple effect, right? So that means the little leg part has to change its shape. And when you get up here, it causes the rear foot that's bound to ADP to swing forward, right? And you're like, what? That's crazy. Yeah, but that's all just a function of the fact that these are proteins changing their shape to get comfortable, right? So if ATP binds, right, it causes a ripple effect of the protein having to change its shape. So this evolved. So it's the right amino acid sequence so that the one in the back has to swing into the front in order to kind of get to a more comfortable conformation. Right? And then once the one swings to the front, it loses its ADP and it gets ATP, right? And so that then becomes the rear foot, and the other foot then swings forward over and over again. Right? So here is an illustration of one of these kinesin molecules attached on one end to this huge cargo, right? So its fancy clamp region is probably clamping on to, this is a vesicle, let's say, with proteins sort of studded in the membrane of the vesicle. There are probably certain proteins that this clamp can be the right shape to grab onto. And then here are the 2 feet walking along a microtubule, right? And so of course you're like, I want to see this actually work, right? Yeah, so I wouldn't just leave you kind of hanging like that, right? So this is an animation. Check this out. How crazy is that, right? So as the foot that's in the front binds to ATP, right, it causes the rear foot to swing in front. Then it cleaves the ATP to ADP, so it swings back up and binds another ATP, right? Over and over and over again. Now, this is a massive vesicle of some kind. You see the texture of the surface. These are probably phospholipids, right? So you can really see the fluid nature of this membrane, right? You can almost see it rippling as all these phospholipids that make up the membrane are just held in place there, right, by the fact that this whole thing's surrounded by water. And so the hydrophilic and hydrophobic nature of the phospholipids are defining this membrane. Now, keep in mind, it wouldn't just be one kinesin molecule working here. It'd be a whole series of them, right? All attached here, moving along. Right? So how quickly can this thing move? Okay. So it's been clocked that a typical kinesin molecule like this can bind ATP and convert it to ADP about 80 molecules per second. 80 ATPs per second. That means the rate at which these feet are moving, the steps, are 80 steps per second, right? So if this is a cartoon, they draw it like a cartoon where it's like a blur, right? But let's think about that. How rapid is that? When we think of our cell as this complicated dynamic place with all of these extensive highways and roadways inside of it, that hopefully after this lecture, you have a greater appreciation of, right? That you don't usually see those when people are looking at organelles, but if you actually focused on that extensive network, how quickly are things moved through the cell? So you can calculate this. If a typical ATP takes 80 steps per second, right? Because that's how quickly it can catalyze, or a typical kinesin can catalyze ATP at 80 molecules per second. Each step the actual size of one step is about 80 angstroms, right? So that means you're moving at about, so 80 ATPs per step times each step is 80 angstroms. So you'd end up moving about 6,400 angstroms per second, right? You're like, what's that? That's like about 640 nanometers per second, right? And if a typical cell in your body is, let's just say, 6.4 micrometers, right? That means that these little kinesin molecules can move cargo from one end of the cell to the other end of the cell in like about 10 seconds, all right? So if you were to blow this up to the size of our island, and you think about all the highways that are on the island, all the little roads, and all of these little trucks and vehicles are cargo being moved around on these roads, right? Getting across one end of the cell to the other in 10 seconds, you know, the scale of this would be kind of appropriate, right? You'd have a truck being about the size of one of these protein complexes. That's like you getting in this truck in Hawaii Kai and making it up to Haleiwa on the North Shore in 10 seconds, all right? That's how amazingly efficient these things are. And it's incredible to think that these things happen. They move almost in a comical way, like these cartoons, just because they are molecules that evolve to be the right shape so that when things happen to them and interact, right, they can carry out these remarkably complex functions that themselves almost makes it seem like they are alive. Right? Okay, so that's it for today. So hopefully you have a new appreciation for this sort of underappreciated part of the cell that we call the cytoskeleton, right? And definitely some of the most beautiful images of living cells are taken by actually highlighting and looking at these different types of cytoskeletal components. Right? All right, let's go ahead and stop there.